Good afternoon, everyone. I'm Noriaki Hashimoto, a member of this driven medical science team. So today, our team invited Professor Thomas Walter from France and have a talk by him titled Predictive Models in Computational Pathology. Please let me introduce him briefly. So he's a professor at Mainz, Paris, and director of the Center for Computational Biology. And his research focuses on the development and application of methods in machine learning and computer vision to biomedical, biological, and medical image analysis. So he received his PhD degree at the Center for Mathematical Morphology at Mainz, Paris, in the field of computer-aided diagnosis of diabetic retinopathy. He then joined the Edinburgh lab at the European Molecular Biology Laboratory. So in 2012, he joined the Center for Computational Biology at Mainz, Paris, first as an independent researcher and since 2018 as director. So his work focuses on bioimage informatics with applications to high-content screening and computational pathology. Since 2019, he holds a chair at the Paris Artificial Intelligence Research Institute, and he is a full professor of Mainz Paris since 2021. So he is also active in teaching deep learning for bioimage analysis in a number of courses, and he has published more than 80 articles, highly cited. Okay, so Professor Walter, could you start your talk?
Yeah, thank you very much for this kind introduction and also for the kind invitation. I'm really honored to speak here in front of you. And so I will mainly talk about computational pathology, but I wanted to take a couple of minutes just to introduce generally the topics that we are working on. So that is a group at École des Mines, as Dr. Hashimoto has already said. So École des Mines is an engineering university. So we want to work actually on biology and biological data, right? So we bring in this mathematical expertise, but we also have to, of course, to have a strong partner. And our strong partner is Institut Curie, which is a hospital and a research center dedicated to cancer. Yeah, so the rationale of our group is to analyze and integrate the massive and heterogeneous data that is really characteristic today for biology and medicine, and to develop specific machine learning and artificial intelligence methods on this in order to do three things: to answer questions in fundamental science, to help discovering new drugs, and to contribute to the precision medicine paradigm. And in precision medicine, we actually want to find the right treatment for the right patient, right? And of course, at the very heart of this is a prediction task. A prediction task where we want to make a prediction at the patient level from data that has been acquired from the patient. And this data traditionally came always from the molecular scale, molecular characterization, single point mutations and gene expression and so on. And this led then to the discovery of many biomarkers, and this is still a very active and very interesting field. However, this is also a very complicated thing to do for several reasons. One reason is that this is a very high-dimensional problem, and the other reason is that we actually try here to make a prediction across 10 orders of magnitude and kind of bypass many intermediate layers, which are themselves also important. So in a way, we throw away a lot of information. So in the end, one way to go would be really to be interested also in these intermediate scales, like the cellular scale and the tissue scale. And here, images are actually the most informative and most important source of information. And this could ideally complement then molecular characterizations. So the role of images is really to bridge this gap of 10 orders of magnitude, but also to provide very complementary pieces of information like morphological phenotypes, the temporal dimension, and the spatial distribution of cells inside the tissue and of molecules inside the cell. Now, I just will show a few applications that we have been involved in before really starting with my main topic. So one way how we can connect the molecular scale with the cellular scale is actually by loss of function phenotyping. So in loss of function phenotyping, what you simply do is you would downregulate or knock out one particular gene at a time, and then look at what happens to the cell if that particular RNA is no longer possible. And this was something that we have been working on in 2010. Unfortunately, this is going to be always in the way, I have the impression. Okay, yeah, I can't put it somewhere where it doesn't turn. Okay, whatever. So I can send you the PDF afterwards, then you get the references. Yeah, and so what we did here was actually to downregulate every RNA in the genome, and then we recorded for 48 hours the phenotypic response of cells in this particular condition. And so this, of course, was asking for computational methods in order to analyze these data. At that time, that was now nearly 15 years ago, we still did this with handcrafted features and support vector machines. So we segmented all the nuclei, we represented each nucleus with a vector of handcrafted features, and then we trained a classifier support vector machines in order to assign to each nucleus at each time point one out of several predefined morphological classes, and then putting everything together, we ended then up in these multi-dimensional time series that we see here at the left, at the right, and from these time series, which represent the percentages of nuclei in each of these predefined morphological classes, we can then identify those genes for which the phenotypic response really deviates from normal, and then we can do some experimental validation. Of course, you can do many more things. You can use the time series in order to provide clustering. You can do hidden Markov models for error correction and fate analysis in these time-resolved data. You can also do ODE modeling and trajectory analysis in order to find movement patterns, and this we did in a series of publications in a couple of years ago. Another interesting thing that we can look at is not only the what happens if an RNA is absent, we can also look at what happens if an RNA is present, and precisely we can look at where exactly it is in the cell. This also provides a lot of functional information, and the which can also be done at a very large scale. So we can do this for many hundreds and thousands of RNAs. And yeah, and so this we have been working on for a number of years now also. So the relevant technique here is single molecule fluorescence in situ hybridization, that's SMFISH. And so what you see here is basically a cell, and each of these dots here is one particular transcript. And you can now be interested in where do they localize in the cell. For this, we found handcrafted features and also some deep learning technology that allows us to tell different localization patterns apart from each other and allow us to identify also new localization patterns, and also link this then to mechanisms in the control of gene expression. And again, this was a series of publications that we in which we have published these results. An important point here is that if you go here really for deep learning versus traditional machine learning, then you always have the problem that you do not have enough data to learn from. And so here we developed also a framework where we use simulated data for learning these features, and that works to some extent relatively robustly. And then perhaps the last point I wanted to mention before really starting with the pathology is that today we have one new modality that has been invented like 4 years ago, which is spatial transcriptomics. In spatial transcriptomics, what you can do, you do this single molecule FISH is to say you label all the different RNAs, but this time not in a cell, but actually in an entire tissue. And you can repeat this process with a certain barcoding scheme that allows you to visualize hundreds of RNAs at the same time for the entire tissue. And this we show here. Now, this time not for hundreds of markers, but only for 7. And this is a piece of lung tissue. We see this white here. This corresponds to the nuclei. And these individual points here, these are individual RNAs that have been labeled by fluorescence in situ hybridization with a certain scheme. And so here, that's really a very interesting field for everybody who wants to do computer vision, because it's challenging data. It's pretty original data because it's point clouds. And so what you have to do first is actually to segment the cells. And you see already from this whitish signal here that this is a challenging task. But if you look now at the points, then you know also that the transcription profiles between cells, they can actually change. And if you use that type of information, then you can develop methods in order to assign each RNA to one particular cell and to segment cells then in using this piece of information. I'm not going to go more into details, so we just have just came up with one method, because I really want to concentrate now on the pathology part, but it's clearly a very interesting type of data that is that requires also a lot of methodological development, and this makes it also so different, so interesting for engineering students, right? Okay, let's go now to the tissue scale. And if we go to the tissue scale and we want to build predictive models, then what we can do is we can predict something about the patient, like overall survival or progression-free survival, or for instance, response to treatment. But we can also go into the other direction and actually predict some genetic features or transcriptional features from the tissue. So why would you do that actually? Because ultimately we are interested in the patient. Well, there are several reasons. First of all, we just want to know what is predictable. We want to know whether there is a strong link between a tissue phenotype and a particular mutation, or between a particular expression profile and the tissue phenotype, right? So that's interesting just from a scientific perspective. But there is more to that. You can also use this type of prediction scheme in order to find a surrogate marker. So often we have already some genetic markers for a certain treatment response, or something is known already at that level. But it's expensive and logistically challenging to actually acquire this. And for this reason, the using H&E scans, using H&E images for this makes this actually feasible. Okay, yeah. So this I wanted to say just in order to motivate that it's interesting to make predictions from tissues, but tissues are challenging data. Why is that? In the end, it's just images, and we know that computer vision and artificial intelligence work so well on images. I mean, we have seen so much progress in this. Well, one of the main reasons I would say is that they are so big and so complex. There's so much going on. There's so much information inside. And just from a technical aspect, yeah, it's just a science also, right? Here we see a breast cancer slide. Yeah, so if you compare that to, and we see 4 zoom-ins, right? So different regions of this tissue, and so these zoom-ins, they are typical images that can be processed by a GPU and by a normal neural network, right? But if you look at, but the images that you have, so one single whole slide image corresponds actually to 50,000 of these smaller images. And what makes things worse is that the label that you have, if you want to make a prediction at the patient level, is not at the tissue, is not at this small image scale, but it's actually for the entire whole slide image. Yeah, and for this reason, people have come up with a technique which is called multiple instance learning. There are different flavors to this, and I just take you through this rapidly, and then I go more into detail. So the idea here is that we cut the whole slide image into small tiles. Each of these tiles is then encoded by a first neural network that's parameterized by θ1, right? From these tile representations are then agglomerated by an agglomeration function, which can be parameterized by θ2. And then that brings us then to a slide representation. And from the slide representation, we can then train a network in an ordinary fashion in order to predict the output variable. So let's just talk about the tile embedder, because there much has happened recently. So what we did traditionally was actually to use some pre-training, which you probably very well know. So you just pre-train an ImageNet, and then in the end, what you take as a representation for this image is actually an internal representation prior to the classification or output, or let's say the last, just downstream of the last convolutional layer. So this is a little bit in the scheme lost here. Okay. Yeah, and so it's a little bit surprising that the representation that can be used for classifying wombats and zebras is actually also useful for histopathology, but it works actually surprisingly well. However, we can do better. And one of the major inventions, I would say, for computational pathology was really the invention in the early 2020s of self-supervision. And again, there are many different flavors to that, but just in principle, we start from the original images, from the tile images, and then we would transform them in different ways. You know, we draw a transformation from a transformation space, and then we have pairs of images that come that originate from the same original tile, right? And then we train a neural network in such a way that tiles that come from different original images are far apart from each other, and tiles that are just different with respect to the transformation, so that come from the same original image, are actually close together. And this brings you not usually pretty powerful representations that can be, and that are very generic, and most importantly, for which you have not used any label, right? Any annotation label. You just, you know, it's really unsupervised with respect to that. Yeah, and so there are many different flavors and recent developments also regarding this. So there's still a lot of movement in this. So it's not yet very saturated from my point of view. The next part is really the agglomeration. And again, we have many different options. We can just take the average. That seems a little bit dumb as a method, but it actually works often surprisingly well and should always be included as a baseline. But you could also project each of these tile representations to a score, and then just take the maximum score, or just take the few maximal, so the k top scores and the k bottom scores in order to represent the tile. But what most people do today is actually what we call attention multiple instance learning, and this is shown here. So you basically represent your slide representation here is a weighted sum of the tile representations. And these weights are actually predicted by a neural network. So that's a typical attention mechanism, and that allows the network to concentrate on specific parts of the whole slide image, whereas to neglect some others. Yeah, and the rest, of course, this here is trivial. It's just a few layers of neural network. Okay, so yeah, but this I would now go to some applications. So that's the general framework. And the first is about automatic grading. And so this is now in the field of computer-aided diagnosis. automatic grading is one of the major applications in computational pathology, and it's about identifying kind of a grade, severity of the cancer. And in this particular project, we wanted to automatically determine the grade of uterine cervix. That was data from a challenge that was held in France. It was about 1,000 slides, with each was labeled with a grade 0, 1, 2, 3. And we had roughly 6,000 annotated lesions, because this grade, and that's very fortunate and normally not the case, but here it's the case. So when the whole slide level is corresponds to grade 3, we also can have a lesion that corresponds to the same grade. So the challenge is now, how can we combine these slide level and the tile level annotations, and how can we also weight the error, because we know that making errors between different classification results, between different grades, has not necessarily impact. So this is a little bit how it looks like, what we see here in these images. So this is a normal tissue in this cartoon. So here we see already a low grade, so that's grade 1. And we see these little halos here around some of the nuclei that appear. These are actually infected cells. They're infected by a virus, and this is the typical phenotype. Then there's low grade, so you lose the structure, this nice gradient here. This is used and lost, and the nuclei, they become also bigger, and then you have cervical cancer where the structure, the tissue is completely lost. Yeah, and so these errors have very different impacts. Of course, if I say it's benign and in reality it's carcinoma, then you could actually argue, so that's a very hard, I mean, it's a very bad result. It's a very bad error. Whereas whether it's low grade or high grade, even specialists, they're not 100% sure. So in this sense, it's actually logical to also weight errors differently according to which errors you make. So in this sense, it is not exactly a classification task, right? There's not a classification task, it's not completely mutually exclusive, and there's also some ordinal character of the grades, and you have these different costs. And so for this, what you can do is actually to define a cost matrix that tells you how bad your error was really between what you said and between what is actually the truth. And here, this is a cost matrix that was given by the organizers. And so the question is, how do you make training now in this particular context? And so our idea here was to use something that has been developed by others, which was, still to implement this in this challenge here, which is a smooth one-sided regression loss that actually tries to regress on these vectors here, on the row vectors here, right? Instead of actually doing the classification, but not with an L2 loss, but with this particular loss, that basically what the sense of this is that if for the classes, when you do a mistake and you are above 0.3, then this is actually not so much penalized as if you were lower. And for the main diagonal elements, it's just the opposite. Okay, yeah, so the next question was, how can we use both the self-supervised, so both the whole slides labels and the tile level labels? And so this is what we did. We started with unlabeled tiles, and we first trained a network with the self-supervised learning paradigm. We did so because we were thinking that in general, self-supervision gives you a lot of benefits with respect to full supervision even. And we actually tested both, and we actually could show that this was really the case. So we started with the self-supervision, and then we trained a network using the loss that I've just showed before, and the self-supervised loss, so a combination of these two losses, in order to fine-tune this network we had found. And then we plugged this network here into the multiple attention-based multiple instance learning framework, and we then fine-tuned this using the one-sided regression loss. And when we then tested this, then we found out that, yeah, so different scenarios, then we found out, like many others did at the time, that the ImageNet encodings, they kind of work, but they are really clearly outperformed by self-supervision. We also found that supervision is actually not better than self-supervision plus full supervision. That was also an interesting finding, and so we ended up then to show that our combined score worked actually better than the competing scenarios, than the competing setups. What is perhaps more important is that these feature representations, they have more of a biological meaning. It's perhaps a little bit daring to say that, but I show you just briefly how we tried to figure this kind of things out. So all of these encoders that we have seen, they provide you the fertile encoding, right? And we were asking ourselves, well, using this tile encoding, what is the sense of these features actually that we get there? And so, first of all, we tried to identify the most informative features, right? Like by a logistic regression approach. And then we took those features that were most informative and we applied activation maximization. In activation maximization, that's a technique where you would find the image that maximizes any given score in your neural network. So you generate an image that maximizes the score, which is kind of a little bit the opposite of what you're normally doing. And so here we found now, well, I'm showing you here now the result. So if we took the most informative feature for the normal person, then we see the image that has been generated in order to maximize this most informative feature. And so what you actually see here is that you have a certain pattern of the input image that is kind of repeated all over the place. There's no global coherence, right? But apparently this pattern here of aligning nuclei, this was something the network has concentrated on. And as it really tries to as shown by this activation maximization procedure here. If you go to the low grade, what you see is actually these koilocytes here. So these are cells that have been infected by a virus, which is normally the base of, the reason for the uterine cancer. And what you see then, that this activation maximization image actually puts these koilocytes all over the place, right? So there are many ways how to visualize and get insights into what happens inside a neural network, but this is one of them, and I have not seen this very often, and I just wanted to share this with you. So in this case, in any case, it actually gave us some useful information. And what I found also interesting is if you do the same thing with different encodings, then you clearly see that that not all encodings, even though maybe they provide a decent accuracy in the end, they do not have necessarily a representation which is very interpretable. And so if you go, for instance, for the image encoding, so this is the most striking here. This image has just nothing to do with an histopathology image. So you don't even see cells or anything. So it's just something different. It's not so surprising. ImageNet has never been trained on histopathology data, right? It's just been fine-tuned a bit, but yeah. And so if you go to a self-supervised learning, what you see is that you have these nuclei all over the place. So apparently the network learns what a nucleus is, right? That's interesting, but you do not have any distinctive feature. And the mixed scenario here was actually that worked best also in terms of interpretability. Another important aspect is, so we are engineers, right? We like having classification tasks, the data, and then we train many models and we ultimately find the best, right? That's our job, right? But ultimately, when you think about it, this is only part of the journey, right? Because after that, you have to think, okay, so let's say I have 80% accuracy. What do I do with this now? Let's say I have 90% accuracy. What do I do with this now? How do I integrate this, right? And so I think we are going to see many publications that target precisely this question also. And this is not only our realm. It's not only up to us to think about this. It's also about people who organize the hospital services, of course, the medical doctors, but also people who have more of an idea of what people would like to, in which scenario they would like to work, right? And so here we tested a particular, well, a very similar grading tool, a little bit in this with this idea in mind. This was now in head and neck squamous carcinoma. So again, we trained a model for automatic grading on roughly 2,000 slides from 456 patients. We got a pretty good AUC, and we also developed a confidence score that actually tells us whether the model was pretty sure about its assessment or not. And we presented this now on a test set of 115 slides to 8 reviewers, to 3 expert pathologists and 3 pathologists and 2 residents. So people really from different levels of experience, right? And when you look at the result, what is funny is that, so that was compared then to a gold standard that was really achieved by experts sitting together and discussing a lot. So we assume that this is right. I mean, of course, this can be questioned also. It's very difficult to really get a ground truth, but I think it's as good as it gets here. And so what is interesting is we had 2 scenarios here. We had one scenario where the reviewers actually saw the AI results, and another scenario where they didn't see the results. The images were completely shuffled, and the entire procedure was not done on the same day, but on several days, with several weeks in between. So you really have to develop a good test protocol in order to make this this work. And so that's now the result. So what we generally see is that most of the reviewers, they actually improve. Not all of them. The experts in particular, they do not improve so much, or they don't improve at all, actually, because they also don't really take AI into account, and they are anyway already at the top level. But many others, in particular for residents, this actually works pretty well. And so that's perhaps some food for thought. How we could use an artificial intelligence in clinical practice. Because one thing is sure, if you have an AUC of 0.9, it's never going to be used at that level completely autonomously. So that's very unlikely. So we have to think about other scenarios. Okay, so that is what I wanted to say about automatic grading. So this is really something, you know, which is clinical, right? But now let's move now more to a research question and to the prediction of homologous recombination deficiency. So that's a defect in the DNA repair pathway that leads to very high genomic instability. So this HRD exists in triple-negative breast cancers and also in luminal breast cancers. And what is most important here, it's a predictive marker for treatment with PARP inhibitors. So that's a useful treatment of breast cancer and in ovarian cancer. Right? So if we can predict HRD, we could identify those patients that would be sensitized to treatment with PARP inhibitors, which actually giving PARP inhibitors would be a good thing to do. Yeah, and so in principle, HRD can be detected by sequencing approaches, but that's very difficult to do for all breast cancers. As you may know, breast cancer is a very frequent cancer. And so that would really impose very serious problems to the national health systems. And so our proposition here is actually to predict HRD by the analysis of whole slide images. So for this, we built a data set of 800 breast cancer patients with known status in homologous recombination deficiency, 309 deficient and proficient and 507 deficient. We also had two subtypes, and then we also had an acquisition over 25 years. And so I'm saying this now because we will see later that this is actually coming with some problems I would make you aware of. That's a typical thing that you actually have very old slides, and this can lead to problems when you do the training. So we trained an attention-based multiple instance learning framework, just as the one that I have just been showing at the beginning of my presentation. And with this, we achieved then a ROC curve, and we can then calculate the area under the curve, and this is 0.88 in our case. But so we were very happy because HRD is something that was not predicted, so it was not known whether it was predictable from whole slide images when we started the project, right? So we really tried blindly to predict something which a pathologist could not do, right? Here, the ground truth really came from the genetic information, right? So that was interesting that we were able to do that. But then we noted that there was a problem in the data. Because if you acquire data over 25 years, that means that maybe your experimental protocol has changed, impregnation, fixation, maybe your dyes have faded out a little bit. In principle, that's okay, right? You have variability in the input data. The problem is, but you always have to check whether this variability actually is linked to the output variables. So that's biases, and in biases, we have more or less this scenario here. We have one discriminative feature and one variable that is not discriminative at all, right? And when you train a classifier, then you would get something like this here, right? But the bias actually appears if you do not have access to the full data distribution, but you actually have access to a subsample of the data, but which is not uniformly subsampled, which is subsampled in some weird way. And if you then train a classifier, then you will actually be able to, then you would actually use a variable that is not at all useful, that is just useful because of the composition of your training set, right? And you can think about this. I mean, there are many different examples that can come from different hospitals, different scanners, and in our case, it was the age of the slide, and related to this, a different experimental protocol to actually obtain it, right? And what you have to check is not whether you have variability in your data, which is perfectly okay, right? But whether this variability is actually linked to the output variable. And if that's the case, and you have thought about everything, then that's not so harmful, because what we can always do is strategic sampling. We build mini batches, and these mini batches, they are sampled in such a way, strategically, that there is no confounding effect anymore, right? This does not completely remove the bias, but it alleviates the bias to a large degree. So if we did, when we then eventually did this, we actually obtained an AUC of 0.86, so it was slightly lower, but this time now unbiased with respect to the technical biases. We then turned to visualization in order to identify also the morphological patterns that corresponded to this output variable, to homologous recombination deficiency. And so here you can do now other things, because this is not at the time level, it's at the slide level. And the traditional way of doing visualization is actually looking at the scores. The attention scores, they indicate clearly the importance of a tile for the decision, right? Because if that's 0, the tile does not influence the slide representation at all. And in this case, you can say, okay, I didn't look at you, so you're not important, right? But and otherwise you have used it, so you know that it's somehow important. However, the scores, they are not actually sufficient to understand whether it's really linked to HRD or HRP. So which output variable you're really referring, this really refers to. And so what we actually did was to use, in addition to these attention scores, also an estimation of the single tile posterior probability. And so this can be estimated by taking the tile representation and entering it at that level. And this is basically a probability that you would have if the entire slide was just made of this one tile, right? And if you take now these two together, so you just look at those tiles that have a high attention scores and those tiles that had a pretty strong posterior probability, so that were closely linked to HRD or HRP, then you can get this kind of visualizations. So this is a UMAP representation of the SSL encodings. So the encodings that we used here for clustering, they are not at all used directly for classification, but classification, and so the label information is just used to filter out those that were either not attended or that where the network was not sure of. So what we ultimately get at is a number of different clusters. And these clusters, they correspond to different morphologies, and they are entirely linked by construction to either HRD or HRP, as we can see here. And so we found some usual suspects like necrosis and tumor infiltrating lymphocytes, but we also found some new morphologies here, like laminated fibrosis, and this kind of indicates that really a hypothesis that we had some teams in our institute had before, which is really the role of the tumor microenvironment, which seems to be really fundamental for many different aspects of the cancer. Okay, so that's one part of visualization. Another idea we had was actually to go more downstream in the network and to take the last layer before we really classify and to interpret this as an HRDness, so as a score for HRD, because it's really very close to the classification. So in this case, the UMAP that we are looking at here is not at all on unsupervised encodings. It's actually something which is more downstream in the neural network. And what you can see is that HRD is actually linked not only to one particular morphology, but to a number of morphologies, and that there's some radial information, some radial distortion of the tissue. So there are many samples in between, basically. And so for this reason, it's a relatively complicated thing to predict. It's not really localized in one particular tile. This is not something that you can expect. Okay, so I'm coming now to our latest development in the field of computational pathology, which is called GigaSSL. And it starts from an observation that was made in a seminal paper in 2019, where a team in the US, the Fuchs lab, they did attention. No, it was not attention, but it was another MIL, to another multiple instance learning strategy, and they wanted to just predict whether the sample contains metastasis or not. So, which is a pretty simple task, right? But they had 40,000 slides, right? So that's a huge number. And so what they did is actually they looked at what is the validation error for different sizes in the training set. And so what you see is that if you are in the 100s here, then you get actually 30% validation error, which is pretty large. And I'm pretty sure if you use self-supervised learning today and more modern techniques, I mean, it's several years old now, you can do better. But still, it clearly tells you that you're in that range here of 1%. You don't do that well, right? And then very rapidly, if you are at 1,000, then you're doing already pretty well. Of course, you go still lower the more data you put into, but you have the impression that here you are really on a very good situation. So the problem is that this is not realistic because 40,000 slides. Who has 40,000 slides? Yeah, sometimes for some cancers and some scenarios, but normally that's not the situation. So we, for instance, we worked on adenocortical tumor subtyping. There we have 55 slides, a rare disease. And these are nationwide in France. And of course, you can get 100 someday, but you will never reach thousands, right? It's a rare disease. Yeah, so that's the situation. But then on the other hand, we have very large data sets without the detailed information. We have the TCGA with 11,000 whole slide images, which are all paraffin embedded. Then we have the hospital routine. We are fully digital at Institut Curie, so we actually generate thousands of slides every year, right? Many thousands, right? So can we take advantage of this unlabeled data? And if we go back now to our initial framework, we have seen already that self-supervision is actually providing an excellent way of making use of unlabeled data, right? But it's always at the tile level. We were asking ourselves, can we also take advantage of the unlabeled data at the slide level, right? Can we kind of learn in a way an agglomeration function by self-supervision? And here's the idea that we have developed here on this. So we have this whole slide image that is decomposed of several tiles, right? And we now generate augmented views of this whole slide image. And here we just have to define properly what we mean by, so we have to find good transformations, and so there can be color transformation, geometric transformation. But what is very important is that we also have samples, subsampling as a perturbation, right? So we work on different tiles. These tiles are then encoded with usual self-supervision like Moco or SimCLR or whatever you want to put here, right? And then they are put back on the map. So we keep the coordinates basically. So we generate a sparse map that is constructed from these tile encodings, right? And on this here, this is basically a sparse image. And on the sparse image, I can now train so a network with sparse convolutions, right? And if I do this, I end up by mapping the slides to representations that are two representations. And now I can require exactly in the same way as before that the representations that come from the same whole slide image should become closer, and the representations that come from different slides should be far from each other. I mean, if we put that together, that means that for any slide that we have then, when we have trained this network, we can map this to a feature vector of 256 features. And what we did now in the following, we just trained a linear classifier on that, logistic regression. Now we tackled a number of different questions from the TCGA, like subtyping question, molecular profiling, and breast cancer. And we wanted to ask, when we compared that then to competing methods. And here DeepMill, this is kind of really what I have shown before. This is really the attention multiple instance learning approach. And so what we see is actually that in most cases, the GigaSSL approach is gives roughly the same. I mean, in most cases, it's actually even a little bit better, right? But not much better. I mean, it's roughly the same result, right? But then we actually looked at what happens if we reduce the training set, the size of the training set. And there we see, so what we see here is now the difference between GigaSSL and the attention multiple instance learning approach. And so we see here what the difference in AUC points is. And so we see that the smaller the data set is, the bigger this difference actually is. So for smaller data sets, it's actually very interesting to work on these unsupervised slide representations. And then we turned to another publication by Carter et al., which was a seminal publication. It's actually not 2023, but 2020. Sorry for the mistake. So where his team actually developed used attention multiple instance learning to predict mutations, individual gene mutations in the genome, right? And we checked, can we predict this as well as they did, right? And of course, with the corresponding test that you have to perform. So we did perform a t-test and then Benjamini and Hochberg correction for multiple testing. And then ultimately, and this is then the result that we actually obtained. We see here in blue what we achieved, and in green what was achieved with the multiple instance learning approach. You actually see that you get many genes. So you see that you can actually predict many more genes with the methods that we have developed here. Yeah, so if we summarize this result for the GigaSSL, we see that we get superior results on small data sets and roughly the same results on normal-sized data sets. We also did an encoding of the entire TCGA, so that's roughly 23 megabytes. The initial data set was 60 terabytes. So if somebody is working on, mutational signatures and subgroups and whatever, you can now use these encodings and actually get a good representation for the histopathology data. You can't relate this to the images anymore. If you want to do that, you have to do deep learning and work on the 16 terabytes. But if you want to do a test, right, then you can do this. You can work on this. And again, I want to mention that we actually did not sacrifice any classification error, right? So I think it's a useful resource, which is of course freely available as a resource. And of course, it's also about computational efficiency. We know that AI also has a terrible carbon fingerprint, and here linear classifiers. I mean, once it's trained, the linear classifiers operating we train linear classifiers that are operating on the representation, and so training such a classifier takes couple of seconds on the ordinary laptop. So that's also an interesting feature of this. Okay, so with this I come to the conclusion. I have talked a lot about first images and then more particular computational pathology, particular learning from weakly supervised data, which is the main strategy today for the analysis of pathology data. I would not like to hide, nevertheless, that there is another alternative way of formulating the problem, which I have not touched here, but we are also working on, which is actually to segment all the nuclei, to classify each single nucleus, and then to try to handcraft features from spatial statistics and to relate them to several outputs. This sounds horribly complicated. It is a huge investment, but the advantage is, of course, that you then get really interpretable features, and for some applications, this is really what you want to have, right? But yeah, so I have not yet something to show on this, so still work on, and we in any way also continue the weakly supervised approaches. Currently, we can predict genetic signatures from whole slide images. That's an interesting thing to mention, in particular homologous recombination deficiency, which also has some clinical value. We need to be careful with respect to biases. It's you can tackle this with many different methods. And yes, the most important thing, the most difficult thing, is that you are aware of their existence. And for this, you have to know your data. And so communication with the pathology departments is really crucial here, right? It's important also then to look what your network has actually learned. This helps you to find errors, but it can also reveal some interesting scientific findings. And then ultimately, I also have shown the GISSL approach, which provides slide level pre-training and a resource that might come in very handy if you do not want to go through the trouble of working on the 16 terabytes of histopathology data. Yeah, with this, I would like to say thanks, and perhaps just acknowledge my funding, and also in particular the two people who have done most of the work I have been showing today. This Tristan Lazare and Mélanie Loubrano, both have defended their PhD end of last year. Thank you.
Thank you for great presentation. So now we can accept some questions from both online and offline participants.
So for online participants, if you have questions, just unmute and ask questions directly.
Okay. So thank you very much for the nice presentation. My name is Islam Rashid from the University of Hugo, and I have a question about the augmented data. So you showed some data where augmented images like changing the color and orientation are included to improve the self-supervised training. So, would you comment on the to what extent augmented data would lead to better training? As in some cases, adding large variability of the data may lose a focus and does not help so much. So, from your experience, how we can adjust the limit of that? Thank you.
So do you mean in the self-supervised framework? Yeah, I mean, for self-supervision, of course, the for self-supervision, the augmentation is of course key, because this is kind of what you would like to revert. I mean, this is not really the general case for self-supervision, what I'm showing here, right? And I said that, okay, we have augmented the data, right? But so what we have to do here is really to augment it very heavily. The idea here of data augmentation is to make the task difficult, right? And so what we train here, what we train the network on, it's not data augmentation like in image classification, where you just kind of virtually increase the data set by augmenting, you know, by adding points that are not exactly the same points, but actually but are next to them in this in some latent space. But here the idea is really to define a pretext task. So it's slightly different. It's also data augmentation, but it's slightly different, right? And it's different, because what you want to do here is actually you define this pretext task by these augmentations, and then you train a network that will put the representations that come from different images apart, and that puts together the representations that come from the same image. You know, and so here, what I can elaborate a little bit on this. So here, what is really excruciatingly important is that these augmentations, they are very heavy, and they can be really outside the usual data distribution that you would expect. What is really important is that it's difficult, right? Because if it's not difficult, if it's too easy, then the network, well, it might learn how to put together things that are already pretty similar, and this representation will not help you in then transferring to other tasks. So here the data augmentation is really central. So what we did in the GISSL approach was to define transformations that are that work on sets of images and not only on images. right? And one of the most important augmentations there is actually the sampling. So we effectively look at different subsets of tiles. Does this answer the question?
Yeah, thank you very much. Thank you.
Okay. Yeah. So we have a question in the chat box. Moti, can you speak up? So if not, I will repeat the question. So the question is, have you thought of using MAE in your SSL strategy? Any opinion on it?
MAE.
Yeah, MAE. I don't know what it is.
I also MAE.
Or something. I don't. Yeah, we have another message. Mask autoencoder.
So we didn't use ourselves. No, we didn't use autoencoders at all. So on this, we have other projects which are more on cell biology, where we have variational autoencoders and masked autoencoders we have not yet worked on. But in principle, it's a good suggestion, right? Because masking has worked very nicely on languages and also on vision. Also in vision, so it's an interesting thing. I must say that what we have shown here, what I show here, this is just an overview slide. Then you have, of course, the concrete implementation, which were not done by us, and so I didn't focus on them. You have SimCell Airmokut, which are the two big ones, but then there's Biol, and now actually in pathology we have some others also. That are a little bit more complex and quite more complex, which use already transformer-based techniques, and in particular, they use a different construction for the positive samples. So here, the positive samples, right? They're always kind of, I take the same image and I transform it twice. But of course, it can happen that the image that is, and this perhaps is very, very closely related to your question, right? There can be another image which is, just next to it, which basically has the same content, right? And if you just do random sampling of your images, you would actually say this is a negative one, but actually it's not negative, right? Because it's so closely related. And so there have been some work where they say, okay, well, maybe we can use very similar images also as positives in the SimCLR framework or in some other framework which is closely related, so for contrastive learning. And so that relates a little bit to this, because masking is also doing this, right? Kind of taking out one tile and predicting it. So loosely related, yeah. Okay, yeah, but there are other techniques which are related to, but which are extensions of what I've been showing here at the time level.
Thank you so much. So yeah. So any other question online? So then may I ask one question? So that activation maximization looks very interesting. So can I understand that you did that for a kind of explanation?
Yeah, it's not an explanation that is meant to be given to a pathologist, right? Because a pathologist will not. You give this to a pathologist and say, like, come on, what's that?
Did you talk with pathologists?
No. Yeah, we showed them. They find this amusing, but they would not like to see that for a real application. No, it's more for us actually. It's more for us actually to understand what are these features that are inside this vector? What do they correspond to? And that's one way of putting it. I mean, there are many different ways. What we didn't show here, for instance, what we also usually do is some blood count, for instance. But I found it original because I had not seen this before, right? And it was. In this case, in any case, it was really striking. We really saw phenotypes that were known to be related to the grade, right? And for this reason, I found it interesting. But of course, it can always happen that in the end, it's an uninterpretable image that comes out. We know perfectly well that the representations that we obtain, they're not always interpretable. and the network takes the decision differently than we would do this, right? So yeah, so I don't know how generalizable this is, yeah, but here it worked pretty well.
Thanks so much.
Yeah.
So are there any other questions online? So if not, we're gonna close disconnect Zoom now. But Thomas, could you share your face if you don't mind? Yeah, Thomas is here. So let's close this session by thanking again to Thomas for a wonderful talk.
Yeah.
Thank you so much.